Good evening again, everybody, and a very warm welcome to today's webinar. Um, we have Dr. Ritika and Dr. Suhas with us who will conduct the webinar. If you have any question and answers, please type them out in the Q&A section, and at the end of the webinar, we will definitely answer them. So we're incredibly honored to have Dr. Ritika Mukherjee and Mr. Suhas Tambe leading our session today. Dr. Mukherjee's distinguished background sets the stage for a truly insightful discussion. She holds a PhD in bioinorganic chemistry from the Yale University and completed her postdoctoral research affiliated with Massachusetts General Hospital and Harvard Medical School. An accomplished author with 17 publications in high impact international journals, Ritika is a respected voice in the scientific community, having presented her work at numerous global conferences. Her expertise is central to drug discovery, covering everything from physicochemical profiling and metabolism to complex drug-drug interaction studies. Notably, she has played a pivotal role in elucidating metabolite structures and mapping metabolic pathways for over 70 novel drug candidates. Also with us today is Mr. Tambe, who is a distinguished pharmacologist by training from the MS University, Baroda. And he brings with him an impressive 20 years of experience across the drug development landscape and the CRO industry. His expertise is exceptionally broad, spanning multiple crucial therapeutic areas, including metabolic, cardiovascular, fibrosis, gastrointestinal, and ocular disorders. Suhas is well known for his proven capability in developing customized in vivo models tailored precisely to project requirements. Throughout his career, he has successfully collaborated on numerous drug discovery programs targeting diverse mechanisms with leading pharmaceutical companies. He has also authored more than 10 publications in high impact peer reviewed international journals. We are fortunate to have both Ritika and Suhas share their deep insights with us today. Over to Ritika now.
Hi everyone, thank you for joining us. Today on the occasion of the World Diabetes Day, which is on the 14th of November, we thought we'll talk a little bit about the recent advances in the therapeutic area and also majorly the GLP-1 GIP platform. So just a second, the next slide. So the talk today is divided into a few sections. The first section, we'll talk about the market, the target, and also the FDA-approved drugs. We'll then discuss the whatever assays, in vitro assays that are required for screening and development of new GLP-1 arrays. And then we'll talk about the in vivo studies that would be required to develop these different therapeutics. So to begin with, let's think about this problem of diabetes and obesity, how big of a problem that is for the world. If you look at the statistics here, the global prevalence of diabetes is supposed to increase to 853 million people by the end of 2050. And that is in the age group of 20 to 79 years. So this will be a huge global impact on the healthcare, and it's going to cost around $2.1 trillion at in the year 2030 to cater to this huge healthcare challenge. If you look at obesity, by the year 2030, we will be at 25% of the people in the world would be facing obesity challenges. So that is 1/4 of the world population, and that would cause a large, again, economic impact where $4.32 trillion will be spent annually on this disease by the year 2035. So the current treatments that are available, which include metformin, insulin, and other hyperglycemic drugs, work well for the disease. However, with such a large population having to deal with these diseases, we need to come up with some superior treatments which have less side effects and also are more effective. And that is where the glucagon-like peptide 1 and glucose-dependent insulinotropic polypeptides treatments have come into the picture. So what is GLP-1 and GIP? So these are incretin hormones that are produced in the intestine when we eat food, basically. And GLP-1 is a 30 amino acid peptide, which is produced in the distal ileum and colon. And GIP is a 4 amino acid peptide. It's a small peptide, which is produced in the K cells in the duodenum and the jejunum. When we eat food, our body these hormones stimulate the insulin secretion whenever we eat food because of the rise in the blood sugar levels. And the receptor for GLP-1 is present all through the body, whether it is the brain, the liver, or the fat tissue, kidneys, or the heart. So, what is the main function of these hormones? First is they help to increase insulin secretion. So they stimulate the pancreas, the beta cells in the pancreas, increase the insulin secretion whenever the blood sugar is high, that is when we eat the food, and help to lower the blood glucose levels. They also reduce the glucagon release. So glucagon is a hormone which actually raises the blood sugar. So they help to reduce the glucagon release. And they promote satiety. So basically, they tell the hypothalamus that this feeling of fullness will come that you and they suppress the appetite based on that. And the fourth function is to slow the gastric emptying time. So there is a delay in emptying the stomach. So you feel full and you reduce the food intake. However, these natural hormones, these GLP-1, GIP, are suffering from degradation issues in the body, and they have a very short half-life. So our body has peptides, which proteolytic enzymes, which can actually chew up these peptides, and one of these is DPP4. So to cater to this problem, scientists have come up with GLP-1 mimics, which behave like the natural hormone GLP-1, bind the same receptor, and carry similar functions. So now these GLP receptor agonists, GLP-1 RAs in short, help in lowering the blood glucose level, especially in type 2 diabetes, and cause significant reduction in the HbA1c levels. They're also now especially used in weight management for obesity, and they suppress the appetite and enhance the satiety feeling by acting on the hypothalamus. They also have cardioprotective effects. They reduce the total cholesterol. They improve the blood pressure and help to reduce any cardiovascular events that happen. Since this GLP-1 receptor is found throughout the body, they also have an effect on kidney health and also on neuro diseases. So currently there are some trials underway where people are looking into using GLP-1 RAs for Alzheimer's disease as well. There is a huge market for these GLP-1 RAs, and current market size is about 62.2 billion. And it's supposed to increase to about 157.5 billion by the year 2035. But the major chunk by the year 2035 would be catering to the obesity market. So the current drugs, which are for weight loss management, weight management would be more important by that time, because you've seen that 1/4 of the population may face this challenge by that time. So what are the current FDA approved drugs, and which particular disease they are catering to? So if you look at it, the first drug that came into the picture was exenatide. Basically, exenatide is a non-human peptide which was isolated from the Gila monster saliva, and it was approved in the year 2005. This is a very short acting peptide, and it has a half-life of around 2 to 3 hours when it is given subcutaneously. So it required that the patient has to take it twice daily. Because it required a lot more effort for the patient, they decided to come up with a longer acting one, which could be given once weekly, which was a synthetic exenatide, basically. However, again, there still it was not as effective as now the drugs that we have in the picture. So the next drug that came into the picture was liraglutide in the year 2010, and this was again a subcutaneous injection once daily, but they had increased the half-life to about 15 to 18 hours, and the fact that this was a GLP-1 analog. It was a modified GLP-1 analog. It helped in reducing immune responses which were observed in the case of exenatide. So this was the next drug. And then came albiglutide, dulaglutide, and lixisenatide. These were all longer acting drugs, which were given only once weekly as a subcutaneous injection. So this reduced the pressure on the patient, and also adherence to the dose regimen was much better in the case of patients when the once weekly injections came into the picture. The most commonly used currently is semaglutide, which is also a GLP-1 analog, and it is again a once weekly injection, subcutaneous injection. It is one of the most effective ones in this class of compounds, which are all single agonists, RNs. And there is another oral version of this Ozempic, which is called Rybelsus, and it's an oral drug which was approved in the year 2019, and it is given once daily. Now, the most recent drugs are the tirzepatide formulations, which are Mounjaro and Zepbound. These are actually dual agonists. So not only do they work on GLP-1, but also on GIP, the other incretin hormone, and receptors. So if you see, there are three which I have colored here. One is Saxenda, then the Wegovy and Zepbound. These three have been approved for weight management, and these are all once weekly injections. Basically, it's very similar to the ones that are approved for type 2 diabetes. However, the doses given for weight management are a bit higher. So when we go to the next slide, I've separated these drugs categorized them into different categories based on one is the mechanism of action. So liraglutide, semaglutide, exenatide, all are GLP-1 RAs. They work only on the GLP-1 receptors. Then we have the dual receptor agonist, which is tirzepatide, which is now more effective than the single receptor agonist because it can work on both the hormone receptors at the same time, GLP-1 and GIP. And then there is retatrutide, which is under development currently, which also, along with GLP-1 and GIP, works on the glucagon receptor. So this is potentially going to be more effective treatment. The most important thing for these GLP-1 RAs is the dosing regimen and how fast or how slow do they act. And that actually determines the dosing regimen. So the short-acting peptides like the exenatide and liraglutide, they reduce the gastric emptying time. So their main function is to reduce the gastric emptying time. However, if you think about semaglutide, tirzepatide, which are all once weekly injections, subcutaneous injections, they are better at lowering the glucose effect. So that is why they work better and they circulate longer in the body and cause a long time effect. There's only one oral daily tablet, which is the Ribelsus. Again, that is the semaglutide version of that. So now these are all very effective drugs. However, there is still a lot of work that is in progress and needs to be done to optimize these for to prepare better versions of it. And what are the properties that you would need to look at? So the first property that needs to be looked at is the receptor binding and efficacy. So in the case of tirzepatide, which works on both GLP-1 and GIP, we need to look at both the receptors. But today's focus, we are looking at GLP-1 RA, so we'll talk about that here. And this definitely affects the dose because you need to achieve potency. It's again structure dependent. So where generally all these are GLP-1 analogs. So which amino acid you are connecting the linker to, fatty acid chain to, how long that is. It will all affect the lipophilicity of the compound and then affect the potency of the compound. The next thing which are related to each other are the frequency of dosing and the dose concentration. They both depend on the albumin binding of the drug. So basically, all these RAs are typically high albumin binding drugs, and the stronger their binding to albumin, they will be long circulating in the body. So the frequency of dosing will reduce. However, if they are too strongly bound to albumin, they may not be available for creating the effect that they are being given for. So that is why the dose concentration and the frequency of dosing will be optimized based on the albumin binding. The other thing that is very important is the DPP4 degradation. So of course, that was a bigger challenge in the case of the natural hormone, but it is still something we need to look at when we are actually developing new GLP-1 analogs. All these GLP-1 analogs have less immune responses because their structure is similar to our naturally occurring hormone. However, this is something still needs to be tested in the beginning to understand that by changing the structure too much, are we changing the immunogenicity profile? So liraglutide and semaglutide are not very different from the natural hormone. That is why the immunogenicity caused by them is very limited. So that is why that is not a concern in their case. But however, when we are working with new GLP-1 RAs, we really need to make sure that that is not going to be a concern. So if I go into a little bit more detail on these assays, the first assay I'd like to talk about is the receptor binding and potency determination for the GLP-1 RAs. So this assay, in vitro assay, is conducted in CHO cells or in BHK cells, which overexpress the GLP-1 receptor. When an agonist binds to this receptor, activates it, it stimulates the adenylate cyclase and in turn releases cyclic AMP. These cyclic AMP levels which are increased are measured using immune cells. If you look at the first table that is given here, just a second, I'll try to get my pointer. If you look at the first table that is given here, the first row talks about the natural GLP-1. And you look at the potency of the natural GLP-1 in this assay is about 0.019 nanomolar. However, for the liraglutide, it is 0.15. And in this case, it's written as NNC01130217, actually semaglutide. And for that, the potency is about 0.16. So there is still a lot of room for improvement. So the natural hormone is still almost 10 times better than the current drugs in the market. This table actually shows, this is from a publication and shows that while the development of semaglutide was going on, they have tried many different linkages, many different fatty acids, diacids, and different sequence modifications and acylations. And what they have observed that semaglutide was the best in that class of compounds, and it in their assay the potency was about 6.2 picomolar. So different assays will give different potencies, but these are the different receptor binding assays that can be conducted. And based on the fatty acid length or the lipophilicity of the compound, final lipophilicity of the compound, the receptor potency will change. At our facility at Eurofins Alpinas also, we perform this assay in the CHO cell line, and the method is developed and validated for using the RLD and the cell-based assay kits. The next assay that I would like to go to is the protein binding assay. Like I said in the beginning, that it is very important to understand the protein binding of these drugs, because the binding dictates the dosing regimen and also the dose concentration. So like we know, liraglutide is a once daily and semaglutide is a once weekly, because it can circulate longer in the body. And for it to circulate longer and be more stable in the body, it binds to albumin quite strongly. However, doing a plasma protein binding assay with these analogs is a bit challenging, because they are lipophilic and have strong non-specific binding to all surfaces. We have developed at our facility this equilibrium dialysis assay, where we have optimized our assay using plasma and even human serum albumin, and reduce the non-specific binding. Again, because of the non-specific binding, performing LCM-SMS analysis also is quite challenging with these molecules, because they bind to pretty much all the surfaces, including the tubing and the instrument. So by including a few modifications in our method, we were able to achieve an LLOQ of 2 nanogram per ml. And with a range of 2 to 5,000, we have conducted a number of plasma protein binding studies for these molecules. And all of them pretty much show very high binding, more than 99% binding to human serum albumin. So the other thing that we need to really look at is the drug-drug interaction liability. So even though these are all peptides and are not metabolized by CYP isozymes, they do have a possibility of causing drug-drug interactions. And because many of them are small, so they may actually even get metabolized by CYPs. So not just by metabolism-based drug-drug interaction liability, they also have these absorption-related drug-drug interaction liability, because they slow the gastric emptying time, because of which the Cmax and the Tmax of many of the drugs is affected. But here I'm going to talk about more about the liability related to metabolism. So we need to perform CYP inhibition and time-dependent CYP inhibition studies in human liver microsomes with these peptides. The requirement is very similar to as is required for a small molecule. We determine the IC50 in the case of CYP inhibition study, and in the case of time-dependent inhibition, we see if any binding of this molecule or the peptide to the CYP, will that cause result in reduction in the metabolism of any other compound. Similarly, for CYP induction assay, it is performed in cryopreserved human hepatocytes, and we determine we perform this using for 3 CYP isozymes, CYP1A2, 2B6, and 3A4, and determine the mRNA expression at the end of the assay, where we actually incubated for about 72 hours. Then the last in vitro assay that definitely needs to be performed is a DPP4 degradation assay. So the GLP-1 natural hormone is degraded by this enzyme by chopping off two amino acids at a time when you have a penultimate proline at the end terminus. So the way we can stop this is by first we could block the end terminus, use some unnatural amino acids, and also we can use circular peptides. So these are the ways we can actually stop this degradation. The assay that is performed requires incubation of the test item with this enzyme at 37 degrees, and then collecting samples at different time points to observe the whether it is metabolized, what is the percent remaining. And what has been observed as is that semaglutide is the most stable of the GLP-1 analogs currently in the market, and less than that is liraglutide, and of course GLP-1 is a very quickly metabolized hormone. So these are the assays that are required to be performed in the early stages and also in the development of GLP-1 RAs. But beyond that, we need to actually look at the in vivo studies as well, including the efficacy screening studies. So for that, I'm going to over to you, Suhas, and he will take you through these screening models.
Yeah, thank you, Ritika, for taking us very widely about what GLP-1 is, what is the market size, how it is being used, what are the molecules currently approved, which were starting from the development of this therapeutic area and receptor agonist, as well as what are the current approved drugs which are being widely used. And actually, these are the hottest molecules right now in the market. And thank you, Anjali, also for the brief introduction about us and saying good words about us. So with that, we'll start the second part of this presentation for what are the relevant screening models, in vivo screening models to test the efficacy of these molecules, as well as if there are any newer molecules which can be screened. The reason for in vivo screening of these molecules is very important because the models that can be used for testing any of the molecules or the GLP-1 agonist should be relevant, should be easily developed, and it should also feature the same features that are present in humans, if not all, at least majority of them. And the most and the last and the most important, I would say, is there has to be translation from mouse model or rat model, animal model to humans. So that way, in this particular therapeutic area, I would say these models that we are going to discuss today are very much important, very relevant, and there is a lot of translation from mouse model to human, which mimic very closely to human disease. So for this GLP-1 therapeutic area, now the trend is increasing with addition of more and more mechanisms into one molecule. For example, it started with only GLP-1, then was added with GIP, that is tirzepatide. And along with that, now the third one is coming, GLP-1, GLP, glucagon. Even people are trying with amylin receptor and few more. So with another 10-15 years, since the market is also growing and newer mechanisms are coming up, it is also important what animal models to choose for how to screen these molecules in these models. So with this, there are three most relevant models which can be used easily and available across the world. First is for earlier screening is for diabetes and insulin resistance would be dietary induced obesity model. Although there are rats, mini pigs, and other models which can be used as DIO or diet induced. But C57 mice fed with 60% high fat diet available with research diet and many other companies is the most relevant and most widely used across the world for screening of these molecules. Not only these, but any molecule which has been screened for diabetes, obesity, or insulin resistance. The another model, it can be either high fat diet or some genetically modified. So single point mutation, there are two models available. One is OBOB mice and DBDB mice. OBOB mice with leptin gene modification and mutation, and DBDB mice, which is leptin receptor gene mutation. OBOB mice develops only obesity and insulin resistance, and it does not get into frank diabetes. Whereas DBDB mice goes very similar to the human disease development for diabetes. Initially with low glucose levels and very high insulin levels with setting up insulin resistance. And over a period of 10 to 12 weeks, it goes with the insulin dropping of insulin levels and parallelly increasing glucose levels. And by the end of 18 to 20 weeks, they develop frank diabetes or become, I would say, type 1 diabetes. So along with that, other than these molecules, there is also growing interest in developing small molecules for GLP-1 receptor agonist, which is very rare and uncommon, but still Pfizer has developed one molecule, liraglutide, to understand if these small molecules can actually go and bind to human receptors. So people have also developed GLP-1 or humanized mouse. They are available with Genove, and we do have access to these mice, where you can screen your molecule, and you should be sure if these molecules show effect in these. So there is going to be a good translation into human, since these are humanized one. So we'll take you during the presentation to one by one model how these molecules are screened, what are the effects of these molecules on various parameters which are relevant for metabolic disease, obesity, and diabetes, and then conclude this presentation. So the first and most widely used model is diet-induced obese mice, which can be used as anti-diabetic screening or anti-obesity screening. And the popular one is with fed 60% kilocalorie fat diet from research diet. From 6 weeks to 8 weeks also, it starts showing increase in obesity. But by 10 to 14 weeks, you can see there is drastic difference between control diet fed or normal chow diet fed animal versus this high fat diet fed. So about 25 gram mouse normal chow diet versus 40 to 50 gram mouse for the high fat diet fed mouse. So the utility of this model is it shows hyperglycemia, it develops obesity, it shows glucose intolerance, it shows insulin resistance or insulin tolerance. So all of these parameters can be screened. And along with that, it also shows metabolic derangement, what is seen in the human, that it changes in triglyceride levels, free fatty acid levels, cholesterol levels, even liver function tape and function test and other biochemical parameters, which can be relevant for the therapeutic area that can be screened. So all in all, this DIO model, similar to human, shows similar metabolic derangement, increase in obesity, insulin resistance, and metabolic parameters. So these model typically we use at Adgyl with 10 to 14 weeks of diet, and we can screen with different treatment. There can be different treatment groups at different dose levels. We can perform weekly blood glucose levels. We can also look for daily body weight changes and food consumption, because these are the main mechanisms for GLP-1 RA. If required, we can also perform, people have performed OGTT. Then you can analyze various biochemical parameters. You can take tissue histopathology, weights, also analyze various biomarkers which are relevant for metabolic changes. So if you see semaglutide, which is now the most widely used across the world for diabetes as well as obesity under two different brand names with different doses. But semaglutide in DI mice, when they are fed for almost 10 to 14 weeks, you start treatment shows a clear dose response starting from 1 nanomole per kg per day, 200 nanomole up to 100 nanomole per kg. So beyond that, there is no need to go for. Already you see almost 20 to 30% reduction in the body weight. You can also see that there is significant drop immediately in the food intake in these mice. So that significant drop in these mice is the main mechanism for initial drop in the body weight. However, as we go on with the treatment, there is again increase in the food intake, although it is much lesser than the control animals, but still there is increase in food intake. That can be maybe because of some tachyphylaxis or due to reduction in the effect in gastric emptying or body's compensatory mechanisms. So these mechanisms which still show significant reduction in the body weight is because of central suppression of appetite center, and that continues. So if you look at tirzepatide and semaglutide as a cumulative food intake, there is still significant reduction in the body weight and food intake compared to control animals. So that shows the utility of this DI model for screening GLP-1 GIP RA. Interestingly, if you continue this treatment and stop the treatment, so for any of the treatment, if you stop the treatment, there has to be reversal in the effect that has been shown. Suppose there is some toxicity or signs of any adverse events, you can start the treatment and the person should go back to its original. So with our studies and published literature, when you stop the treatment after say 4 to 6 weeks, immediately these animals start showing increase in body weight. These MRI pictures clearly show the fat. When with treatment there is reduction, and when you stop the treatment again, it starts gaining the body weight. So this means there is no toxicity related effects on this treatment for the reduction of body weight. Now, when you collect at the termination different blood for different parameters, metabolic parameters which are relevant to show that there is metabolic effect, there is significant drop in leptin, there is significant drop in triglyceride levels, free fatty acid levels, liver triglyceride levels. There is significant drop in fasting glucose levels, insulin levels, indicating there is improvement in insulin resistance and fat glucose levels. So all these, all in all, together, reduction in obesity ultimately improves insulin resistance and diabetes symptoms. Also helps in improving cardiovascular function, since there is reduction in triglycerides, cholesterol, and fatty acids. So you can also perform mRNA analysis to show the mechanism by which either for these therapeutic area, or if you are using any combination, or if you are using any maintenance therapy, the UCP1, PGC1 alpha, all these markers are responsible for increase in fatty acid oxidation. So these are from white adipose tissue, and significant reduction with fatty acid binding protein, indicating there is less accumulation of fatty acids into the adipose tissue. Now comes more relevant mouse model. So this therapeutic area or these RAs are approved for both diabetes and obesity. So for diabetes, the most relevant model across the globe would be either DBDB mouse or a similar rat model called ZDF rats. Instead of ZDF rat, people try to use more of DBDB for the reason it requires very less amount of compound, and it is more reliable. And as I explained earlier, this model shows similar phenotype as humans, and the development of diabetes from insulin resistance to diabetes can very well captured within 10 weeks of age in this particular model. So starting from 6 weeks, if you start using, by 18 weeks, you actually end up with having a frank diabetes. So these DBD mouse available with JAX and Harlan can be used very effectively in testing these GLP-1 therapeutic area. So these mice, as I said, they show a mutant. They show they are polyphasic, polydipsic, polyuric. They show high insulin levels in the beginning, and as they grow, they show decrease in pancreatic islets. They show decrease in insulin levels and increase in very high glucose levels going up to 600 to 800 mg per dl. So chronic treatment is widely used in these mice for screening. So if you can actually give treatment for maybe 4 weeks to 8 weeks and measure different parameters like glucose levels and uh, body weight, feed intake. So DBTB mice with liraglutide, tirzepatide, and retatrutide, you can actually visualize there is difference in the effect with lira versus tirzepatide. Tirzepatide being dual, showing the highest effect on fasting blood glucose and body weight change compared to vehicle-treated animals. So these mice, when treated for 4 weeks, they also show significant reduction in HbA1c level. So HbA1c is the marker which shows you your glycemic control over a period of 3 to 4 months, about 120 days, because hemoglobin bind to glycine, RBCs, is reduced, meaning there is long-term glycemic control on in this model. So there is good translation that there is significant reduction in HbA1c percent in these mice that you can also see similar effects in the mice. Here, I would also like to add important observation. Semaglutide or all these molecules have different PK profile in different species. So that's why we have also measured semaglutide levels in the plasma. We have LCMS and methods available for performing and analyzing all these large molecules using LCMS. The reason for showing this is for the translation. In mouse, the half-life for semaglutide, say for example, is about 7 to 8 hours, whereas in dogs and monkeys and mini pigs, it is about 150 to 160 hours. And in human, it is even more. That's why it can be used as once weekly. So this translation is also important to see that what you see with daily dosing is really not required for when you go to higher species. That's why when you're developing at a late stage, you also need to perform at least PK studies to understand the PK profile of these molecules in higher species. So another interesting model is similar to OBOB. This is a DBDB mouse, which is showing spontaneous mutation in leptin gene and not the receptor. And it has obesity, hyperphagia, diabetes-like syndrome, but not diabetes. So it has hyperglycemia and not as much as DBDB mice, but very high elevated insulin levels, showing insulin resistance. And this model can also be used if you're developing your this GLP-1 therapeutic area or any other molecule for obesity. So in this model also, after 8 weeks of treatment at various doses, administered as a subcutaneous single dose daily for several weeks, up to 8 weeks, there is significant and dose-related reduction in body weight. At the same time, as explained earlier, there is initial drop in the feed intake. However, that feed intake then later on, after a week or so treatment, comes back to almost close to the control animals, but as a cumulative feed intake, there is still reduction as compared to control animals. So they show a very good effect in this particular model. Other than this, there are other models also available. People do high-fat fed rats, which becomes actually costly because you have to spend more on the fat diet, and the compound requirement also goes up. So mouse is more widely used for screening. Then now, very recently, MASH, or earlier it was called as NASH. So in the NASH therapeutic area, there was not, for these many years, not a single molecule which was approved for treatment. Resmetirom is a very recent approval, which is a thyroid hormone receptor agonist. And in OBOB mice, if they are fed then glycine and amylin diet, it's called as GAN diet. So OBOB mice fed with high-fat diet plus GAN diet develop features of MASH, or metabolic steatohepatitis. So in this particular model, with semaglutide treatment for about 4 weeks, so development takes about 6 weeks, and 4 weeks of treatment show significant reduction in body weight, which semaglutide has already demonstrated. But the more interesting is there is significant reduction in liver weight as well as the biggest biomarkers, which are ALT and AST for any MASH or NASH or alcoholic steatohepatitis. A very significant reduction or highly significant reduction in ALT AST levels with semaglutide. That is also reflected if you look at the histopathological assessment, the ballooning and reduction in the vacuoles that are formed in form in control diet or which are reduced with semaglutide. So compared to even resmetirom, semaglutide is showing a better efficacy with reduction in triglyceride and cholesterol levels. With histopathology is the most important for this. So all the parameters of fatty liver disease or steatosis score or fibrosis score or the ballooning of hepatocyte is significantly reduced with semaglutide treatment. So more and more molecules from this area, these peptides can be used for development of these therapies, or can be used for MASH, which actually develops into finally liver cirrhosis. So with this, the molecules that we use, the models that we use and background, we'd like to conclude our presentation here. Over to you, Ritika.
So, to conclude, we would like to just say that we discussed today the market, the target, and the mechanism of action of these drugs. Some of them are short-acting, some are long-acting. However, what we have seen for patient adherence, long-acting ones are more favorable. The other thing is that these are not, there is no one answer for all patients. We need to test while giving these therapies. It is to be followed up, and routine assessment by a dietitian and dietitian and all the other doctors has to be done, and ensure that there are no side effects. Because if there are side effects, then that particular therapy needs to be stopped. So there is still a lot of work that needs to be done and can be done to improve the efficacy and the other properties of these RAs, and it is underway, and more will be done. And for that, we at our facility we have relevant in vitro and in vivo assays which can help you develop and screen your molecules. And there are currently, especially, there are a lot of therapies which are adjunctive treatments, like lixisenatide and insulin is an adjunctive treatment. It is in the market, but there is a lot more work that can be done to determine and to have the most effective treatment. So with that, we would like to open.
And additionally, I would also like to add, for any therapy which is effective, will also have some or the other side effect. So recently, there are many concerns and publications coming up for this particular area. Although they are showing very steep and long-term effect in reduction in body weight and diabetes improvement, still, but with as there is reduction in the fat, there is also reduction in the fat mass, which creates a lot of concern among community and even the prescribing physicians. And there are some studies that can be performed to address this if there is any muscle loss, because at late stage, leading to muscle loss is also not beneficial. So these things can also be addressed by performing histopathological assessment of type 1, type 2 fibers, measuring them and then counting them if any of these therapies with long-term treatment has any such side effects. And as scientists, we should be able to address those issues also, by which the novel therapies with devoid of such side effects can come up for the better treatment options.
So with that, we'd like to end the talk. Thank you so much for listening.
And move to the questions. If anyone has any questions, please, you can feel free to type them out in the Q&A section. So Ritika and Suhas, there are a few that have been shared. One is here, which the person wants to know what are the difficulties associated with PK studies of GLP-1 agonists?
So, as
Yeah.
such, there aren't that many difficulties. I think the major challenge is to actually measure the concentration of the, develop a correct LC-MS method, which can actually determine the concentration of the compound in the plasma and tissues if required.
Yes, I agree with Ritika. So there are as such no difficulties. We have at our facility performed several PK studies for all these molecules which are we have presented, and across different species, including rats, mice, dogs, and mini pigs. So as such, I don't see any difficulty. Only method development is a challenge initially, but if you have high-end LCMS, it is easily doable.
There's another one which they want to know what are the other major metabolism pathways for these molecules other than DPP4?
See, these are peptides, so are metabolized mostly by proteolytic enzymes. See, they're not CYP metabolized compounds. So I would say more of the proteolytic enzyme degradation. That is what happens for these molecules.
And then we have one more. How about the determination of immunogenic responses in the mice models you mentioned?
Yeah, even we have a MSD platform. If any of the new molecules which has any immunogenic reaction, there is Luminix assay or MSD platform. We can inject to the mice, collect the blood samples, or even the site of injection and check different release of various cytokines and see if there is immunogenic response. So with the native molecule, which is currently the I would say RLD, there is no immunogenic response. But however, if somebody is developing a new molecule with little modification, even in sequencing the one amino acid may trigger some immunogenic response. That's one. Second, if there is in the synthesis, something is added up in the peptide as an impurity or something, if that has any immunogenic response, but that can be addressed and measured in mouse model with Luminix and MSD platforms available.
Great. I think that's it for now. So thank you so much, everybody, for joining our webinar. Thank you so much, Ritika and Suhas, for your time and valuable insights. I'm sure everyone has gained in-depth knowledge from all of this. And again, thank you, all of you, and have a good night.
Thank you. Thank you so much, everyone. Thank you, Anjali. And thank you,
Thank you.
everyone, for joining this webinar.